Hey guys, Sarah here. So today we're going to be going over how to calculate 3 different corn snake loci, or more easily said, 3 different genes. Now with different loci, there can be multiple genes that share a locus. So this is not going to necessarily be applicable to those genes, but this is if you're calculating 3 basic genes in corn snakes and in pretty much anything else. This refers to, like I said, 3 different loci. So if you've been following this series, you know that a loci is a sort of a spot on the DNA where information for a gene is held. And the allele is what is in the locus. The allele itself has the information for the gene, and then the locus is where the allele sits, essentially. And if you have not been watching this series, this probably looks like a whole bunch of gobbledygook. But if you'd like to learn how to do corn snake genetics from the very basics on what a Punnett square is, how to read it, how to make it, all the way up to what we're doing today and then beyond, I plan to do more of these videos later on, then I'm going to put a card above so you can go back and start at the very first video and just watch the whole playlist if you would like to do that. So I went ahead and did all of this by myself, and I'm going to actually do one with you guys after this that was requested on Facebook. If you would like to be involved in the things that I do in the videos, definitely check out my Facebook page, facebook.com/sarahsnakeshop with periods in between. I'll put it in a link in the description down below so you guys can go easily check that out, but you can vote for the type of videos that I do, what I do in the videos, and stuff like that. So check that out if you'd like to do that. So this is 3 different loci being, I decided on amel, hypo, and diffuse, because those were the ones that the capital letters and the small letters look very different. Now I know that in the past I have not done the capital letter and small letter version. I have used an X to mean that there is no gene, but in this case I wanted to go ahead and start using how a normal biology class would be doing it with the capital letter and small letter version. So a capital letter is going to mean that the gene is there, and then the small letter is going to mean that the gene is not there. And so a small letter, like if there were 2 small letters, like if anywhere you see 2 small a's, that just means that the amel locus is empty. 2 small a's mean that there's 2 sides of the DNA, but neither of them have either of them. Neither of them have the actual gene on there. If you see 2 capital A's, that means that the A locus, the amel locus, has the gene on there. And then obviously if you see 1 capital A and 1 small a sitting next to each other, that means that one side of the DNA does have that gene and the other side does not. So I went ahead and put all that up here for you guys. If you'd like to look at it, you're welcome to pause and kind of take it all in. I am going to erase all of this and then do the one that was requested on my Facebook page as I record so that you guys can do it with me. So if you would like to actually calculate something with me, then go ahead and get out a pen and paper. You want to orient it this way because if you try to orient it portrait style, there's probably not going to be enough space. I do want to explain a little bit why everything's laid out the way it is in case it's confusing for you guys. So obviously I have A, D, and H. So this snake here is a normal that's heifer, amel, diffused, and hypo. And then this snake is a homozygous amel, diffused, and hypo, also known as a hypofire. And these, of course, just list what all that means, the capital A's and small a's, etc. And I went ahead and laid out this way and this way all the different possibilities. Now, in order to get all these different possibilities, you would have had to do like separate Punnett squares for each individual thing. And obviously I did not do that this time, but the easy way to sort of calculate that is to match up each of these. So the front here, or the top here, I'm sorry, is for this parent. And what I did was go, okay, capital A could match up with capital D and capital H, but then capital A and D could match up with small h. So that's where I went. So I have capital D, A, D, H, and then capital A, D, small h. And then I was like, okay, the capital A could be small d, and then large H, which is this, and then capital A, small d, small h. And I went through and did that with all of these. And you would obviously have your small a, large D, large H, etc. And then with this up here, they're all going to look like this since they're all capitalized. But I did want to go ahead and explain this to you so you guys would sort of understand why that is. And I'll kind of draw some lines so maybe it'll make sense. So the first one went A, D, H. And then the second one went A, D, H. And then you end up with like, then this A going to this D and then this H, and then A going to this D and this H, and then your small a is going to go here, here, small a would go to here and then here, and you know, you can kind of get the idea. It would go like there we go. And obviously just trying to draw all that just looks super complicated, so I didn't want to have all that just on the board. It's just a bunch of lines, but kind of so you would understand that each letter on one side is paired with, I don't want to say paired, I guess try, I don't know. They're put together in threes, depending on the different ways that it can all line up. So this capital A, like I said, could line up with this capital D and the capital H. This capital A could line up with the capital D and the small h, etc. You get it. So that's what's up here. And then, of course, this one would be this parent down here. And I went ahead and wrote them all out. Now, I did not have space to write it all out like completely horizontally. I had to pile them on top of each other. So, like, for example, I'll go over here so it's easier. This A, small a, capital D, small h is here. And then I just put the capital A, capital D, capital H underneath it here instead of writing it straight out in this direction because there just wasn't enough space. And you can, it's actually easier, I think, to do it this way because then you can see the pairs right on top of each other instead of having to like go through and find the pairs to put them back together later. So you can see down here I went ahead and just put all the pairs together, and that's easy to do when the letters are like right on top of each other instead of all kind of strung out and you have to go like look for them kind of. So I'm going to go ahead and erase this and then do a really fun one, like I said, that was requested on Facebook. So I'll be right back. Actually, really quick before we do that, I'm going to move the camera closer so you guys can see a little better what everything is down here. So as you can see, we moved everything just sort of down. All of these, like this whole column is all the same all the way down. And these are all your hypo fires. And then this column here is your fires, het hypo. And then all of the columns are the same all the way down, which is where you get your 1 out of 8. So I hope that that makes sense. I kind of just wanted to show you guys all these because I figured maybe you wouldn't be able to read them all from there. So you get your 1/8 hypo fire, 1/8 fire het hypo, 1/8 hypo amyl het diffused, 1/8 amyl het hypo and diffused, 1/8 hypo diffused het amyl, 1/8 diffused het hypo amyl, 1/8 hypo het fire, which is amyl and diffused, and then 1/8 normal that's het for hypo and fire. So that is how that is calculated. So I'm going to go ahead and stop this and do the fun one. Okay, so if you are following along with the pen and paper, we're going to start from scratch. And I know this is a really long one, so just bear with me. So Joseph Blevins was actually the one who suggested I do this. The pairing being gold dust tessera to an amyl het caramel, just to sort of play with it and get a better idea of how calculating this is going to work. So what I'm going to do is assume that this tessera is a het tessera, not a homozygous tessera. That will make it a little bit more fun. A homozygous tessera is going to kind of make everything a little bit more cookie cutter, but I want to make it a challenge. So what we want to do first is actually separate out all the different genes in each snake. So with a gold dust tessera, we have caramel tessera and amyl ultra. So the tessera is actually going to be, I'm going to do capital T and small t since that's our heterozygous tessera. The caramel is going to be the next easy one So we are going to be working with a capital C and a small c, which might make it a little weird, but we will figure out how to work with it. And then we're also going to have AML and Ultra. And since both AML and Ultra are going to be there, I'm going to actually make A and U so that we know exactly which genes we're working with. Because remember, we're working with AML and Ultra, and they are both present on this snake. So over here, we're going to do the AML het caramel. So there's no tessera, but remember we have to put a placeholder in for tessera. So the placeholder is going to be our 2 small t's, and then we have our het caramel. So that's going to be, oops, since this is a homozygous caramel, it needs to have 2 capital C's. My bad. Het caramel is the capital C, small c, and then our homozygous AML. So we have A and A. So what we want to do is make our Punnett square a very large Punnett square, and we want to make it 8 by 8. So we're going to make it very, very large. So we want to leave some room on the sides and some room on the top to actually like write everything, but a good chunk of our paper and or whiteboard is going to be taken up with the square. So what we want to do now is figure out where all of these are going to line up. So we're going to start with our capital T. So our capital T can line up with this C or this A. So I'm going to say capital T, maybe, capital C, and capital A. So that's our very first one. And I'm just going to go ahead and make a line straight down here because that's going to be the column for that. Now, this capital T could pair up with the U instead. So we have our capital T, capital C, and capital U here. And we're going to take that down. Let's see, we have our capital T, and that could also pair with this C here. So we're going to pair it with the second C, even though it doesn't change, we're going to have to put it in anyway. So capital T, and then C, and then A again. T, C, A, and then again, T, C, U. So here, let me go ahead and make the column for that. T, C, U. And the reason that we have 2 that are exactly the same is because this capital T has to pair up, or like 2 sets that are the same, is because this capital T has to pair up with both of these C's and then A and U. So that's why. It would be different if like one of these 2 C's was different, then one of these sets would be different. But since the C's are the same, these 2 sets are going to be the same. And we're going to have a similar thing going on with our small t here. So the small t can pair with this C and then our A. So we're going to go with our small t, capital C, capital A. Put that here. Small t, capital C, and then U, T, C, U. And I know that I make my small u's, or my large u's look like small u's. Just remember that if there's a U, it's there. I always have made my u's this way ever since I was really little, so just gonna have to bear with me on that. That's not a habit I'm gonna change just for this video. Okay, and then we do it again. Small t paired with this C, small t, C, and then the A. Whoop. And then small t, C, and then U. And U. Alright, so that is 8, and obviously I have all this extra room over here. I'm just going to shorten up the square since we don't need all that space. We might be happy that we had some space on the side anyway, because later on I kind of want to have a space to write and like let you know what's going on with everything. So we have 1, 2, 3, 4, 5, 6, 7, 8, which is what we were supposed to have. We're supposed to have an 8 by 8. So now we're going to do this on this side with the no tessera het caramel visual AML. So we're going to have this small t is going to be paired with this C or this A. So we have small t, capital C, capital A. So I'm going to actually move over here to do this. I know that I might be out of the frame right now, but it's easier for me to write this way. C and then A. And I'm going to go ahead and make a row here. And let's see, so then this can be paired with that. And then the second A, so these are going to be the same, small t, big C, big A. I'm going to do that. All right, let's see. So our first, our first T then can pair with the small c and a big A. Excuse me. The air is so dry in here. I know that I complain about that in a lot of my videos, and it's like, Sarah, why haven't you just gotten a humidifier? And the answer is, I don't know. All right, so we're going to do the same here, small t, small c, and then the second big A. All right, I'm going to go ahead and continue filling this out on my own, and then we'll come back. Okay, so as you can see, I went ahead and finished filling out the second half by myself, and most of the reason for that is just because the second half is pretty much the same as the first half. I think it is actually exactly the same as the first half, because we ended off, I think, at the first 4, and then the second 4 is exactly like the first 4. And that is because, again, we have these 2 T's, and each of the T's has to be paired with everything all at once. Or not all at once, but like individually, separated like this. So I'm going to go ahead and actually fill out the square, and then we will talk about what each of those things means. So I'm going to start, you can go either way. I'm going to start actually up here and work my way down with everything first, and then we can work our way across with everything else. So I'm going to start with our TCA. So we have capital T, capital C, and capital A. And then our capital T, capital C, and capital U. All of that's this stuff up here, in case you guys didn't notice. T, C, A, T, C, U again. The C's, I'm afraid, are going to confuse us, but I'll do my best. Then we have our small t, capital C, capital A. Small t, capital C, capital U. And then we do that again. T, C, A, T, C, U. And then these can go down. I'm going to probably go ahead and put them down like this. If you are curious as to why I'm doing it this way, you just have to fill out the square. And I'm going to do it on top of each other like I did in the previous one, because it is easier to read, in my opinion, to do it that way. You do not have to do it one on top of the other. You can write it out completely. So I'll give one example of just writing it out like straight across, and I'll just go ahead and do that here. So writing it out straight across, I'm going to start up here with our small t, capital C, capital U, and then you would take small t, capital C, or no, that's a small c. Small c, I knew that the C's were going to be a problem. Small t, small c, capital A. So we'd have T, C, and then A. So that's what it looks like when it's written out straight. If you decide to write one on top of the other, then it ends up looking more like this, T, and then small c, and then capital A. So that's what it looks like when it's written on top of each other, and then this is what it looks like when it's written out completely straight. Either one works, they're both fine. I'm going to do most of them like this though, because it's easier to kind of pair everything up, in my opinion, to get the visuals, to like be able to read the visuals that you're getting. So that's just my opinion. Yeah, so I'm going to just keep going. Like I said, the C's might get confusing, but I'm going to do my best here to make sure that we know that like a capital C is bigger. That is the biggest reason why this whole time I've been using X's instead of the small letters, because a lot of the English alphabet like look the same, capitalized and the small letter, and C is just a really good example of that. P is another one. So I've been avoiding it, but I figured I'd go ahead and I'd just go ahead and do it once for you guys. So you can kind of see maybe why it's confusing. It's also really confusing that when you have a lot of X's just hanging around. So that's one reason that I went ahead and did convert over to using the small letters and the big letters, is because if you just have a ton of X's around, that it also gets confusing as to where those X's are supposed to go. So I'm going to go ahead and fill this out. And you guys can skip if you want to the end of this. I'll put a timestamp in the comments below that you can go click to just like go to the end of this. But if you'd like to follow along, you are welcome to. So all the ones that are on top are going to be the top ones here, and all the ones on the bottom are going to be the side ones. That's kind of how I'm doing it right now. So T, capital C, capital A. Already did that. Small t, capital C, capital U. I need to be giving myself more space. That's another problem I have right now. All right, so small t, capital C, capital U. I did it again. Here, small t, capital C, capital U, small t, capital C, capital U. I also had space at the bottom, which I was surprised about and glad about, because any extra space that we get filling this out is going to be helpful. Let's see. T, C, A, T, C, A. I know this is like the most boring part of this, and I'm sorry about it, but for those who are following along, I know it's probably helpful for you to see everything in action. And if you are still watching this but you're not following along, thank you. I could use the watch minutes. I appreciate it. All right, I'm going to go ahead and do these ones here. So we have small t. I keep not giving myself enough space. C, U, T, C, U. Just got to do it all the way down, and then we got to do all the ones across. And I know this is going to be like an obscenely long video, but I wanted you guys to see the process, so if you ever want to calculate genes on your own, you would know how to do that, because it's actually a pretty useful skill for a corn snake breeder or someone who breeds pretty much anything where you have a lot of genetics going on. It's going to be useful. I'm going to go ahead and just do this half, and then I might kind of move myself over there to do the other half. T, C, A there. And we have T, another one, T. Wait, did I say? Okay, this is actually getting confusing for me because of all the different, like, C's. I'm trying to make sure that I'm doing it right. TCA, TCA, yep, I know, it's very confusing. I'm sorry for anyone who's confused right now. Even I'm a little confused, and it's because of those C's. It's because of those little C's. Would not be so confused if the little C's and the big C's didn't look exactly the same. But it is what it is. Okay, I think that's probably fine. I'm going to actually change this one to the same format as we have everything else, because that'll make it less confusing for me. T, C, A. All right, and up here, right now actually what I'm doing is plugging these in underneath each of these, in case you were confused about what I was doing here. I didn't really announce that I was doing that, so just thought I would mention it for anyone who is trying to follow along, and it's like, what is she doing right now? Because sometimes I even wonder what I'm doing right now, and so I don't blame you. All right, T, small t, capital C, capital A, and that's small t again, capital C, capital A. Okay, small t, small c, capital A, small t, small c, capital A again. All right, small t, capital C, capital A, small t, capital C, capital A. I know that a lot of these are really repetitive, and you're probably wondering why we have to repeat them every time. And the reason is pretty much just for consistency, because if everything was different, like if we didn't have any double T's and we didn't have any double C's, like if there were no repeating genes, each one of these tiles would be completely different. So the reason that we go ahead and just do all of the work is pretty much just to cover all of our bases. Because if we were calculating, like, let's say each locus that we were working with had 4 different allelic genes that could attach to it, and we were working with all of them, every single one of these tiles would ultimately be different. We don't have that, thankfully, but if you ever got to a point where you needed to calculate those things, it's useful to just know how to do this correctly. and to cover all those bases. And what we're doing here is actually statistics. I don't know if anyone has ever taken a statistics class. I did. I took it actually because of corn snake breeding and because I needed a math class to graduate college. And I found it to be very useful. Now, you can do a lot of this like with a calculator and you don't have to like do it by hand. The reason I'm doing all these videos is just to sort of teach you guys like how to do these things, because I know how to do them, but not everyone else does, and not very many people are actually good at teaching it. It's a very difficult thing to learn and therefore a very difficult thing to teach. I've always figured that the harder a thing is to learn, the harder it is to teach. So I wanted to, since I already knew how to do it, since I already learned how to do it, I wanted to pass that on to anyone who also wanted to know how to do it. And I know that I'm not the most perfect teacher, and you guys probably haven't been watching the majority of this, but if you have, like, I just wanted to like do everything I could to help people learn this stuff. I am, however, I think going to pause the video and fill out the rest of this by myself, because I kind of have to like be in front of the camera anyway. And then I'll make sure that you guys have ample time to like look it over and make sure that your square is filled out correctly. Okay, as you can see, I have gone ahead and filled everything else out. It got kind of complicated again, so I actually went ahead and decided to start drawing my capital C's a little bit differently just for this video. So all the ones that have these ticks on the top, these are your capital C's. I went ahead and changed that everywhere so that it would be a little easier to like point out. So now what we want to do is we want to go through and read all of these, which I know sounds like super crazy, but it's not going to be quite as hard as you might think. So if you notice, there's going to be different groups, and I have different colors here that maybe would help us. So you have your capital T, small t group that has, all right, so I'm just going to start writing phenotypes up here in this area. So I have capital T with a small t, 2 capital C's, and 2 capital A's. So I'm going to circle in blue all of those. So I have capital T, small t with 2 big T's and 2 big A's. So that's also going to be, let's see, capital T, small t, 2 C's and 2 A's. This is the hardest part for me, is like finding everything. So it looks like this is another one right here. And, excuse me, this one here. And then this one here. Those are all those. I don't know if they are all of them though. So anything that's over on this half of the board is not going to be that, because these have small t's starting over here. So anything that has a capital T and a small t is going to be on this side of the board. And then anything that has 2 capital C's, I think, are going to have to be here and here as well. And so let's see, I think we have some here. So capital T, small t with 2 big C's and 2 big A's here, and then here, and then also here, and here. Okay, so you might notice that we have an 8 by 8, which means there's 64 different options. And here you can see that 8 out of the 64 are this. And this is a het tessera that's visual caramel and visual amel. So this is a butter tessera. I'm just going to say butt tess. And that's going to be 1 out of 8 or 8 out of 64. I'm going to keep it in the 64 range since we have 64 options. And then we can always reduce those down later if we want to. And those are going to be all of the ones like that. Now I'm moving on to the red marker, and we're just going to move down the line. So we have a capital T, small t, with 2 capital C's, and an A and a U. A, U. And so that's going to be this one, and this one, And then it should be this one here, this one here, and then this one, and this one. So again, we have, oh, and these ones also, this one and this one. So again, we're going to have 8 out of 64. This is going to be your ultra milk caramel tessera, so that's another gold dust tessera. So I'm just going to say GDT for gold dust tessera. Again, 8 out of 64. Okay, now I'm going to move on to a different colored marker. I'm going to go with purple now. I have a lot of different markers, but I'm not sure if I have enough. So we'll see what we can do. We're moving along. Okay, so I'm going to go ahead and move on to the next section, which is capital T, small t, let's see, capital C, small c, capital, and 2 capital A's. So this is going to be your T, and then a het caramel, so C, angry C, that's what I've been calling it to myself, and A, so this is an amel tessera het for caramel, so amel tess het for caramel, that's just how I say it. And again, we're going to probably have a very similar outcome. So I'm just going to go ahead and find those. It's going to be this one and this one. Let's see, and I think this one, let's see, yep, this one here, and this one, and let's see, should be these down here, capital C. Okay, so this one, and then this one, and this one. This pen is not very happy right now. So again, we have our our 2 groups of 4, which is going to be 8 out of 64 are that as well. And I'm going to move on to a different color, but I'm pretty sure that I'm going to run out of colors. I have like 2 other colors, I think, over there that I'll have to maybe run over and grab. Okay, so we're going to do a similar thing, only we're going to do the capital T, small t, capital C, small c, and then AU. So that's going to be this one, 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 and this one. And the reason I didn't really have to read those is because we're coming into a pattern here, which is very nice. So this pattern was our capital T, small t, capital C, small c, getting angry, and then our A and U, which is actually an ultramel tessera het caramel, so ultramel tessera het caramel, and again we have 8 out of 64. And it's going to be a very similar thing on the other side. The only difference on the other side is going to be we're not going to have a heterozygous tessera. The only difference is on this side, it's going to be the exact same pattern, but you're going to just have small t's on all of them. Instead of having like a capital T and a small t, you're just going to have all small t's. So the other patterns that you're going to see are these 2 and these 2 will be the same, and then like these 2 and these 2 will be the same, and so on and so forth. And pretty much it's going to be the same 8 out of 64, only we're just removing the word tessera from all of those. I'm not going to go circle all of them, but I will write them down for you and maybe sort of highlight them for you somehow. So we're going to have our, let's see, small t, I forgot to put hold on. I forgot to make these C's angry. I'm going to make sure that they are angry for you guys. There we go. Been giving them angry eyebrows. Boop. Okay. I like to have fun sometimes. All right. So small t, capital C, A, and then we're going to have our small t, capital C, A, U, small t, capital C, small c, A, and then small t, capital C, small c, A, and U. And for those of you who, I mean, that's going to be the same things up here. So you're going to have your butter, your gold dust, your amel, and your ultramel, caramels. Yeah, I'm sorry, without caramel. These are the ones with caramel, these are the ones without. Sometimes you just work with so many letters that you just don't even know what you're writing down anymore. So yeah, you still are going to have your 8 out of 64, which is ultimately going to be 1 out of 8. It's just not separated out in as like easy to read and as pretty of a way as the one that I had earlier, where I could just say, all right, this column is this column is this, or whatever. But you do get the gist, and I'm sure that you can, if you've been following along, sort of see the pattern in everything. Like I said, like every other pair of these is going to be the same. And that's why I also wanted to do different colored markers for you guys, so you could see like where the patterns were going to lay out. It's a lot easier to do this if you're on an Excel spreadsheet, because you can just make the background a different color or make the text a different color, so that those things are easier to like have pop out at you if you're trying to actually just like make an Excel spreadsheet. That's going to be the easiest way to do any of this stuff. I do it on a board because, that's how most of us learn, probably, is on a board. But if you guys have been using Excel spreadsheets, that is going to be the easiest way to actually go through and do this. A pen and paper is fun. I enjoy it. I think it's like, I like to, is this for the same reason that I like to read books on like actual paper as opposed to reading them on a screen. I just like having it in front of me. I like being able to touch it and actually write it down, and that's just more fun for me. So I hope that this video helped you. This is why corn snake genetics are difficult, and this was actually a somewhat simple-ish one in comparison to a lot of different ones. Like, there were a lot of different options. One thing that was nice about this was each set was actually different. So we had our non-tessera and our heterozygous tessera, so those sets were different. We had our het caramel and our caramel, so those sets were different. And then we had an amel versus an ultramel, so those sets were different. So because each of those sets was different, we ended up with a nice breakdown of 8 out of 64 of each different possibility. If any of these had been the same, that would be a different story. Hey guys, sorry about the camera change there. My camera ran out of memory. You know that your video is too long when your camera runs out of memory. However, I just wanted to continue what I was saying, basically, that the reason that we got a nice 1 out of 64 out of every, or sorry, not a 1 out of 64, an 8 out of 64 on everything was just because the pairs lined up so well. If we had 2 ultramels or something like that, it would have been very different. So I just like to play around with all the different things that can be done. Just also remember, this is more like for fun, or for, just saying that you can do it, or proving to yourself that you can do it. There are a lot of sources out there that do this for you. The one that comes to mind right now is one on Morph Market, and I will link it down in the description below if you guys would rather go there to calculate these genes. It is nice to know this because there are some genes that aren't going to show up in certain places. For example, if you were working with the peach gene that I mentioned in my previous video, one of my previous videos. As of recording this right now, it's my most recently uploaded video, but I'm probably going to upload a video like between these two regardless. So if you do have something like peach that's not showing up in Morph Market or on Ian's Vivarium or anything like that, that's going to be a little bit different. But so that in that case, it's nice to know how to do this, or at least to understand the concepts there. But it's not 100% necessary in everything. So anyway, thank you guys for watching. I really appreciate it. Thanks for going on this weird journey with me. I may do some other things in the future if you would like me to. If there are certain pairings that you'd like to see me put up on the board, you're welcome to comment those below. Or like I said, you could do what Josh did and let me know on Facebook some interesting pairings that you'd like me to go over. And they don't have to be simple like this. They can be super complex. They can be, maybe try to not make them more than 3 alleles, but if you do end up with 4 or 5, we can figure out how to calculate those without even having to use a Punnett square. That is also an option. So anyway, again, like I said, thank you for watching. I will see you in the next video. Subscribe if you haven't.